It's a great pleasure and an honor to be here today, to have been invited to give this sort of joint colloquium for physics, mathematics, computer science, and life sciences. This kind of represents what my career has been like, starting as a pure mathematician and moving on into other fields as well, seeing where this pure mathematics can be applied. I feel tremendously honored to have been asked to give this talk, and it's my first time in Brno. I'm very happy to be here and have a chance as well to discover university and the city, which I've really enjoyed so far. I started off in pure mathematics, and if I were giving a lecture in pure mathematics, which I still do sometimes because I continue to do research in the area, after, 20 minutes or so, the board might look something like this. But that's not the point today. The point today is to tell you stories about how we can use the kind of mathematics that I do, which is algebraic topology, to study other areas of mathematics, network science, but also particularly with applications to neuroscience. So many of us are fascinated by the brain, by its immense complexity, by trying to understand how it is that the brain actually works the way it does. How does it produce thought? How do we translate incoming stimuli into actions and reactions and so on? There's a huge amount to understand. This complexity is enormous. And it's so enormous that in order to come anywhere near understanding what's going on, we need to apply what mathematicians like to call a dimension reduction. We like to reduce massively the amount of information with which we're presented. Because here we have, this is just an artist's rendering of a tiny fraction of the complexity of the brain to something that we can actually apprehend, comprehend maybe, and study. And what's remarkable is that even after this kind of massive dimension reduction, there's still a lot that we can say and a lot that we can learn. The point is to look at this from many different points of view and to learn what we can from these different perspectives and to put this knowledge together that we learn from the different perspectives. So the kind of dimension reduction that I've been thinking about over the last 10 years or so is really just thinking about what's the underlying network, what's the underlying graph associated with a network of neurons, where we're going to see that what we do is associate to each neuron simply a node or a vertex in a network or a graph, and a connection between two neurons as being represented by an edge. And then we see whether there are any kind of maybe higher structures that are hidden there, where we're considering interactions not just between two neurons, but maybe between many neurons. And so these are the sorts of questions that we can explore using the tools of algebraic topology, which is my field of research, my original field of research. So as I said, today what we're going to do is see how we can use algebraic topology to understand things in general about the structure of a network and the function, therefore, of the network, and in particular, with applications to neuroscience. Now, topology is not a kind of mathematics that is usually taught in school. We all learn some algebra, we learn some geometry, maybe we learn some probability and statistics, some analysis, but topology is usually not on the menu for people unless they decide to study mathematics. So one thing I'm going to do is start by talking to you about what topology is. So this word cloud up here that you see on the screen is really describing some of the terms that are associated with the notion of topology, the notion of continuity, the notion of deformation, and so on. But let's take a sort of dive deeper into the details. So what is topology? I'm going to give you sort of three different perspectives on topology. The first is that topology is the mathematics of shape. Now, maybe you know this famous joke about the topologist who couldn't tell the difference between a coffee cup and a donut, right? Because in a topologist's eyes, two shapes are the same or equivalent if one of them can be deformed in a continuous manner so that it looks like the other. And by continuous manner, what I mean is that you imagine that your shape is made of something like Play-Doh, and so you can deform it, you can squeeze it and whatever, stretch it. You're not allowed to cut it, because that's not continuous. You're not allowed to glue things together, because that's not a continuous thing to do either. But how can you deform it? And so you're interested in what properties of shapes are preserved in this kind of deformation. In the case of the coffee cup and the donut, what's preserved there is the fact that there's a hole in the middle. And that really turns out to be enough to be able to qualify what surfaces within three dimensions are actually equivalent. Look at the number of holes. But in general, it's much, much more complicated. So topology is also the mathematics of connectivity. how things are connected. And not only just is there a connection between 2 points, as in this sort of directed graph, but how many connections are possible and what's the relationship between them. And so I could say, not even necessarily is there a direct connection between 2 nodes, but is there a path of edges between them? And how many paths are there? And what's the relationship between the paths? And so on. And those are the kinds of questions that topology provides you with tools to answer. And the final important aspect of topology in this context is it's one kind of mathematics that's particularly good about deducing global properties of something, of a shape, from its local constraints. So you have within the neighborhood of some point, you can say, well, I have various constraints on this, and you fit these things together, and you can see what sort of global structure emerges from these kinds of local constraints. Now, topology is not the only kind of mathematics that's good for this. Algebraic geometry is as well. There are others. But topology does provide us with good tools for this. So how do we get from thinking about graphs to thinking about things in topology? And the way I'm going to explain how to do this has to do with thinking about things in terms of subgraphs, smaller pieces. Because when we're thinking about the really big networks that are underlying networks of neurons or big networks of electricity producers and so on, these are huge networks, or the internet, right? Absolutely huge networks. And if we want to try to apprehend the structure of this network or describe it, a good way to do it, or one approach, is to look at small, more easily understandable pieces and study those small pieces of the bigger graph. So that's what we're going to talk about. The idea is that for any type of graph or network, there are various flavors. They can be what we call undirected, in which case you just have a bunch of nodes and you have a bunch of edges and there's no sense of orientation or direction on the edges. Or you can have one that is directed, so you have a direction from A to B or from B to A. Or weighted, which means that you assign some sort of numerical value to the edge, which could be some sort of cost of traversing edge, or something that tells you about the speed at which you can traverse an edge, or something like that. And for each type of graph like that, you say, well, I'm interested in what would be sort of significant sub-pieces of this graph. And I want to study this really big graph in terms of these pieces that have actual meaning in whatever my context is. And we add one more condition that's sort of just to make things work nicely, which is to say, I'm going to look at a family of such graphs such that if I take one of the subgraphs in this family and I remove one of its nodes and all the associated edges, that it's also in this family. So that just is to make some of the mathematics work out more nicely. And then you could just count, right? You could say, I'm going to look at my huge graph, and I'm going to see how many times, for example, a triangle-shaped graph appears in this huge graph. Just count. And I can do that for all the other kinds of graphs that I consider to be significant like this. And I can sort of give a statistical description of my graph this way. This sort of provides me with kind of local information about what the graph looks like. Or I can say, okay, I'm looking at all of these significant subnetworks, but they could be overlapping. They could be overlapping on the intersections. There could be subgraphs at the same time. And you could say, okay, I want some way of characterizing how they overlap. And that will give me a more global picture of the structure of my graph. So that's the kind of thing we do. And then just to point out, it's really important to choose the family of subnetworks you're looking at in a way that's appropriate to whatever your context is. So whether it's neuroscience or some other source providing you with these complex networks, you choose them based on what should be meaningful in your context. So let's think about the case of directed graphs, because this is what is most important in the context of neuroscience. So when we're talking about directed graphs, we're going to consider the following sorts of significant subgraphs. What my colleagues and I call directed n symplices, and what the people here in Brno call transitive tournaments on n+1 vertices. It's the same thing. So it's going to be a subgraph that is complete, which means between every pair of nodes, there is an edge, and there's exactly one edge, in a way that is acyclic. It doesn't create any cycles. There are no paths that cycle around like this. And on n+1 vertices. So the shift between n and n plus 1 has to do with the question of dimension here. So if I think, well, what is a directed zero simplex? So that means I have a graph on one vertex, just a point, there are no edges. And so this is sort of a zero-dimensional thing. This is why we call it a directed zero simplex. A directed one simplex is nothing but a directed edge. So I have two vertices, I have one edge between them. A directed two simplex, now we have three vertices. Every pair of vertices is linked by an edge in a way that does not create a cycle. So if we look at this, we look very well at this graph. There's one vertex from which there are two arrows that emanate, and there's one vertex into which two arrows come. And so we have a source, which is the one from which the arrows emanate. We have a sink, which is the vertex towards which there are two arrows that enter. And we have a little feed-forward piece. So if we imagine sending a signal from the source vertex to the sink vertex, at every node we know exactly where we're supposed to go. There's a sense of orientation that makes sense at every point. We can continue the story to talk about directed three simplices, now with four vertices. We have a sink, we have a source, so the vertex from which three arrows point out. We have a sink, the vertex towards which three arrows point in. And so we always have this well-defined sense of direction for where we're going. And so this is what we're looking at. We'll give them some names like this. And this is what we decide to consider as a family of significant subgraphs. Why were they reasonable to study in the context of something like a network of neurons where we're thinking about a flow of information? Well, because there's this idea that we want information to get from A to B, and so we want it to sort of be fed forward in this manner. And the idea is that with these directed n simplices, the larger n is, the more robust these networks are, because in the case of a zero simplex, if I lose the vertex, it's gone. In the case of the one simplex, if I lose either of the vertices, either of the nodes, there's also no signal transmitted. But for the directed two simplex and directed three simplex, if I happen to lose one of the vertices in the middle, I can still get the signal from A to B. So it's a more robust system for transmission of information. The larger the n, the more robust. So it seemed like something reasonable to study, but this was just pure intuition. So now we could say, okay, well, we could do this sort of local analysis where we count the number of such things. We want to think of sort of the global picture. How do these things fit together? This isn't what we study when we call what's called the directed flag complex. So it's a directed version of something that's well known in graph theory called the flag complex or the click complex. And it works as follows. So suppose we have a directed graph. And one thing we notice is that I have a directed n simplex, so it has n plus 1 vertices. And I can imagine removing one at a time, each of those n plus 1 vertices, and seeing what's left, removing the edges as well. And what's left will be an n minus 1 simplex. So in the case of the directed 2 simplex, for example, if I remove any one of the vertices, so if there I remove the upper left-hand corner, and so we see that we have an edge. Similar if I remove the upper right-hand corner or the target. So this is a case showing that to each directed 2 simplex are associated 3 directed 1 simplices. So this generalizes for all n. And so this gives a way for us to sort of see how all the different directed n simplices in one of these directed graphs fit together to give a description of the global structure of even a very large graph. So let's see information about this. So what do we get in this context? Let's call this thing FLOG for flag. And it's what's called an ordered simplicial complex. That is to say, it's just a collection of lists that are closed under taking sublists. Because anytime this is this business of being closed under eliminating nodes. And so if I write this in mathematical terms, what I'm looking at is a collection of all subgraphs, little g and big G, such that this little g is of the shape of one of these directed n simplices. And this thing is actually graded in the sense I can just look at it one n at a time as well. And this opens the door for me to using all sorts of tools from algebraic topology that I will stay very, very superficial about in what comes. So let's see an easy example. And one that's going to bring out something important. So if we look at this example here of a small directed graph, and we say, okay, what is the associated directed flag complex? So the directed zero simplices, those are just the vertices. The directed 1 simplices, those are just the edges. And then I'm looking for the directed 2 simplices. So in that case, what I'm looking for are the sets of three vertices that are all-to-all connected, but in an acyclic way. And so in the upper left-hand corner, there's one of these triangles that is not a cycle, and so that's going to give me a two-simplex. If I look at the lower half of the square, the lower triangle in the square, that one is actually cycling around. So that's going to give me a cycle. It's not a directed two-simplex. And if I look at the far right-hand triangle, which now has one of these edges we call reciprocal, or some people call it a bygone, where we have an edge going from, say, A to B, and an edge going from B to A, which is entirely allowed in the directed graph complex that I'm thinking about because we have such connections in the brain, then in fact, here we have two different sets of three edges to look at, one of which is going to make a two-simplex, and the other of which is not because it's a cycle. So if we look at what that gives us, if I fill in the directed two-simplices with triangles to indicate them, then what I see is that in this case, let's see, that the cycles in the graph are actually creating sort of cavities or holes, or if you use the language of algebraic topology, homology classes. And so sometimes when people say, but why do you focus on these directed two-simplices or directed simplices in general when they're these interesting cycle structures? It's because this highlights the cycle structures actually as creating interesting geometric structure in the directed flag complex. Okay, so one more example. Here we have directed flag complex. It's coming from a graph that has 6 vertices. So it's giving these two pyramids that are glued together. One on the top, one on the bottom, and 12 edges. And then there are 8 of these directed two-simplices. And so we write this down by saying f0 of g, that's the number of vertices, is 6. f1 of g, that's the number of edges, it's 12. And f2 of g, that's the number of these two-simplices, is 8. But we could also sort of take seriously this calculation of different cavities, or the Betty numbers associated with this. And so what's called beta 0 of 1 of g is the number of connected components, there's just one. Beta 1 is the number of loops, and there's 0 of those. And beta 2 of g is sort of the number of two-dimensional cavities that we have. And this thing is hollow, so we have a two-dimensional cavity, and that's what we get with beta 2. And the cool thing about this is that if you take the alternating sum of either the numbers of faces, there's f0 minus f1 plus f2, or you take beta 0 minus beta 1 plus beta 2, you get the same answer, which is called the Euler characteristic, which is 2. This is some sort of model of the sphere, actually. So this is the kind of calculation we can do with these directed flag complexes. Okay, so that's it for math for the moment. So how are we going to apply this to networks of neurons? So I got a mixed audience here, apparently, that there's people from life science as well who have been sort of subjected to math for the last 10 minutes, and now we get to a bit of biology. So now the mathematicians get subjected to some biology. We need to talk at least a little bit about neurobiology. I need to tell you a little bit about neurons and their very, very basic elements. So a neuron is composed of 3 major parts. So there's this big yellow part in the middle here that is the cell body, also called the soma. And then you've got these sort of finger-like things that are sticking out from this that are called dendrites. So the dendrites are gathering information, gathering signals from other neurons. And then they do some of the information processing, and then it gets transmitted further to the cell body. And what happens is that the voltage on the membrane of the cell body rises as it starts gathering the signal. And when it hits a certain threshold, it may or may not, this is a stochastic thing, it may or may not emit what's called an action potential, or a spike of electricity, along what's called the axon that carries the signal further to other neurons, the dendrites of other neurons. And their connection points between the axon of the first neuron and the dendrites of the second neuron, they're called synapses. And the signal is transmitted across the synapse by neurotransmitters. And again, there's stochastic elements here. Some synapses are more reliable than others. Some are more powerful than others. So there's a lot of subtlety to the transmission of the signal. But this is the basic idea. From the dendrites to the cell body, to the axon, and through the synapse to more dendrites. Now, this is really a cartoon, of course. Real neurons are much more complicated. So, for example, Here is another visualization. This is done by the amazing visualization team at the Blue Brain Project at EPFL. This is giving you a better idea of what two neurons look like. So the two big blobs here are the cell bodies, or the somas, of these neurons. And what are illustrated here is sort of lit up points. Those are supposed to represent the synapses, where you have the transmission of a signal from the axon to the dendrites. So we're talking about chemical synapses here that are really, they act like as valves. The information flows in one direction. So because these are connections with direction, if we want to come up with some good mathematical representation of a network of neurons, we need to take that into account. And so we want to represent the circuit by a directed graph or a digraph. And that's what we're going to do. So the first example I want to talk about goes back to when I started working with the Blue Brain Project, which was roughly 2014 or so. At that time, they were just finishing up the construction of the Blue Brain microcircuit. So the Blue Brain Project is a big project financed by the Swiss government since 2005. I think it's coming to an end this year. The original goal of which was to build a digital reconstruction of the entire human brain. This turned out, as you might not be surprised to find out, to be a little overambitious. The first real digital model they built was for part of the brain of a rat. So they built a digital reconstruction of part of what's called the somatosensory cortex of a rat. So it's kind of like a core sample. And what you see represented here is this sort of core sample of the brain, where the top part, the sort of pinky blue part, that's near the surface of the brain, and going in deeper. So there are basically 6 different layers in the cortex. And so they built this reconstruction. It's roughly 31,000 neurons with roughly 8 million connections among them. And they used input data coming from 5 different rats that were 14 days old when they were unfortunately sacrificed. And data was gathered about the structure of their brains. And so some biological data about what their brain samples looked like having to do with different cell densities and layer thicknesses and things like that was used as input. And the algorithm that was used then to construct the whole circuit was fairly stochastic. And so what they did was create 7 different instantiations, 7 different microcircuits for each of the input data. So there's 5 from these 5 different rats, and there's one sort of average set of data. And so we had 6 sets of parameters, 7 instantiations of each for 42 different circuits. And so it's not just the architecture of the circuit, because what's really interesting is how you get the function out of the circuit. And so the electrophysiology and the biochemistry is built into the circuit. And therefore, you could not just have look at the architecture of it, but also record the activity of the circuit in response to either spontaneous activity, because the neurons there's always some electrical activity coming on, even if there's no incoming input, because your neurons have to be ready to react. Or you could record the activity in response to some stimulus. So you have an evoked activity. So what do we do? Because this thing was actually a digital reconstruction, you know exactly what the positions of the different neurons are. You know exactly which pairs of neurons are connected. So you know exactly the directed graph underlying it. And so you can compute the directed flight complex, or you can calculate the numbers of simplices. And that's what we see here in this picture. So the blue curve here is counting the numbers of simplices of different dimensions in BioM, which is the Blue Brain model. And what we see is that, so it has roughly 8 million edges. And then if you go up, you see that it has roughly 75 million directed two simplices, roughly 60 million directed 3 simplices, roughly 10 million directed 4 simplices, and it even has 7 simplices. So these are 8 neurons that are all connected in this acyclic feed-forward manner. And this was just the existence of such things was surprising to the neuroscientists at first. They didn't expect to find something so highly structured in this network. And the comparisons we did at the time, because numbers in and of themselves are not so interesting, you have to be able to compare them to something to say how meaningful they are, compared them to a number of different fairly elementary control models, such as the green curve, which is just saying, let's make a random graph that has the same number of edges, and to see whether that also has some of these higher structures, so these higher simplices. And you see that yeah, it has a fair number of two simplices, way fewer than the biological network, and then basically dies out after 3. So somehow this, what we're detecting here is some sort of complexity or non-randomness in this biological network, which is reassuring considering that the Swiss government spent well over 100 million francs on building this model. And then there were comparisons to a couple other more biological models, which still didn't quite cut it. And then we went into the lab to see whether, maybe go back here, went into the lab to see whether such things existed in real life. So they're taking actual slices of brain tissue from rats, and we're able to find 2, 3, even 4-dimensional simplices. And then they were just sampling 12 neurons at a time. And then reran the same experiment in silico and saw that, yes, if anything, we're probably underestimating the numbers of such simplices compared to real life. And then you could say, okay, well, we guessed that these simplices should be interesting because they should have some functional feed-forward kind of role. And so what the graph here is showing, which is probably the most complicated graph I'll show today, is the following. So we looked at all of the connected pairs of neurons in the network, every single pair, and looked at the correlation in their electrical activity. So you looked at some sort of either spontaneous or evoked activity and looked at how highly correlated their electrical behavior was. And so the average over the entire network was something like 0.31, something like that. And then the question was, well, if I look at my connection and I say, what is the maximal dimension of a simplex to which this edge belongs? And then as a function of that, look at how highly correlated or not the electrical activity was. And so what's called dimension along the x-axis is precisely that, the maximal dimension of a simplex to which an edge belongs. And we see that as that increases, the correlation, the electrical activity of the neurons also increases. And the different colors correspond to exactly where in the simplex the neurons are. So if we look at the neurons that are closest to the sink, they're the most highly correlated, which is maybe not so surprising because they share the most input. But for all, no matter what the position in the simplex, the correlation increases as dimension increases. So this was just sort of Pearson correlation. We could also do it in information theoretically, and we just say the same kind of result. So there does seem to be a functional role for the simplices, which has been confirmed more recently. So the last point here is one that I think is quite striking. So when we're looking at another argument in favor of thinking about simplices like this, is that they should be saying something truly biological. Now, if you remember, a few slides ago, I said that when they were building this blue brain microcircuit, they had data coming from 5 different rats, and then they had some biological data that was kind of an average of those 5. And they built these different instantiations. And you could say, well, if what we're measuring with topology here has some biological relevance, then we would hope that if we look at 2 instantiations of data coming from the same rat, that they should be topologically similar. And if we have 2 instantiations coming from data of different rats, you would hope they'd be somewhat different. And bingo, it's exactly what turned up. So what we're plotting here is the number of 2-dimensional cavities in this flag complex, Betty 2, for those who know some algebraic topology, versus the number of 3-dimensional cavities in the flag complex. So these numbers are very big. Here it's on the x-axis, we're looking at tens of millions, and on the right axis, we're looking at millions up to 11 million. And what we see is they're color-coded by the rat from which the data was extracted. And then light blue is this average. And what we see is there's a beautiful clustering behavior here. So you really have that instantiations coming from the same biological data are topologically similar. And we see a distinction topologically between those coming from different biological data. So I think that this is another indication that we're on the right track when we look at these simplices. So time has passed. The blue brain microcircuit was published in 2015. That's 9 years ago now. And by 2022, the blue brain team had gone way further. They'd built a model of the entire somatosensory cortex, not just one core sample, but the entire somatosensory cortex minus a part called the barrel cortex, which is of a somewhat different nature. But still, it's a model that has 4.2 million neurons. So this is considerably more than the 31,000 in the previous microcircuit model. And there are 13.2 billion synapses. And one interesting thing, there are a couple of interesting things about this bigger model. One is that there are connections that are both local, so within a distance that's similar to what we had in the case of the microcircuit, and long range that stretch quite far within this entire somatosensory cortex. Another thing is that we actually have some geometry. So when you look at the microcircuit, it was just sort of this cylinder, right, this core sample. But here you can see, when you look at this model of the somatosensory cortex, it has some curvature. It has the heights can vary and so on. And so we could make the same kind of computations, but see how the geometry affects the distributions of the simplices and so on. So I'm not going to talk about that aspect of it now, but only point out that we were wondering whether we would see the same kind of distribution of simplices in this much, much, much bigger model. And it turns out that yes, that we do indeed. If anything, it's even more complex than what we had in the case of the microcircuit. And here they did comparisons with quite a few different models. So if you look on the left-hand side with local simplex counts, so the heaviest green that says local, that really is the circuit itself. You're just looking at local connections, sort of the same distance that you had in the microcircuit. And there you see that's very similar to the curve that we had for the microcircuit. And then did comparisons with various other random graph models, such as stochastic block models, Erdős-Renyi configuration models, and so on. And you see, yes, they have some complexity, but not that much. And then if you add in the long-range simplex counts, so the gray curve on the right-hand side where it says combined, then you see we have simplices even up to dimension 18 and beyond. These are huge simplices that are all connected. And you see that, again, if we look at these, if we compare these to the various random graph models, that there's nowhere near the same degree of complexity. So it was a nice confirmation that what we saw in the version in the microcircuit was generalizes to these much larger networks as well. The next context in which I'd like to talk about using this sort of language has to do with comparison between the brains of mice and the brains of humans. Now, one would, mice being much smaller than humans, of course, they have much smaller brains. And you could say, well, is the difference, the evident difference in intelligence between humans and mice just a matter of the size of the brain? What goes into really making the difference between the behavior that we observe, the enormous behavioral differences that we observe between humans and mice? And I think that we can give some hints of what might be of part of what's going on, that it's not just a question of the size of the brains or even the size of the neurons. So let's look at somewhat an anatomical comparison between mice and humans. Now, here I'm just looking at layers 2 and 3 of the cortex. Remember, there are these 6 layers. And one reason to do so is that layers 2 and 3, in the case of human brains, are what are best known for various reasons having to do with where we can extract neurons more easily. And what one observes when you compare a mouse brain to a human brain is that the mouse neurons are much more tightly packed than the human neurons. So we have if we look at the heights of the various layers, so the heights of the layers are much greater in humans than in mice. We have much bigger brains, but although the neurons are less densely packed. And the distances, so we see this represented in various ways, having to do with the distance, how many we have per cubic millimeter, and so on. And also in the plots on the far right-hand side, we're looking at the distribution of the distances between the neighbors and so on. So basically, there's a pretty big distance between the neurons of different types. So that's already one anatomical difference here. Another has to do with the size and the complexity of the neurons. So neurons, these are much better representations of what neurons actually look like than the cartoon I showed you a while back. You see something about really how complex these shapes are. So here we're not looking at the axons, we're just looking at the dendrites. So these are examples of what we call pyramidal cells, excitatory cells in the brain. They're called pyramidal cells because their cell bodies, their somas, are roughly pyramidal shaped. At the top of the pyramid, you have a dendrite, usually pointing more or less up, which is called the apical dendrite. Then you have a bunch of dendrites that look like a sort of root system around the soma that are called basal dendrites. And we see that the human neurons are much bigger than the mouse neurons, and they're much branchier. And that just in general, there are all sorts of various comparisons we can make between the distributions. So there's a lot more variance in the population of these human neurons than the mouse neurons. And I'm not going to talk about this today, but we actually have developed as well topological tools for really quantifying the differences between these kinds of neurons. So anyway, we can take neurons and say, okay, we see that there are big differences between the neurons of humans and mice. How does this translate into something about the differences between their brains, the differences between the exhibited complexity of behavior between humans and mice? Well, what we can do is build networks of human neurons, using the blue brain machinery, build networks of neurons that are of human type or of mouse type, and then do this same sort of flag complex comparison and say, okay, how many simplices do we have of these various dimensions? And so we see on the left-hand side here is just looking at these pyramidal cells and looking at a network that's built of 10,000 human cells or a network built of 16,000 mouse cells. So you think, well, that should be more complex, right? It has more neurons in it. But in fact, the complexity of the human network is much, much higher than that of the mouse network. Because if you look on the y-axis here, we're taking the logarithm of the number of simplices. So that there's huge orders and orders of magnitude more complex, the human structure than the mouse structure. So that's for the pyramidal cells. And then if you look at cells that are called interneurons, these are inhibitory neurons, there's less of a difference, but there's this huge difference in the case of pyramidal cells. So the hypothesis is that where is this complexity of the network structure coming from? It's coming from the complexity of the neurons themselves. And this compensates for this lower density of the neurons in the brain tissue, and it really giving us this more complex network structure, which one could hypothesize is necessary or for the more complex behavior that one observes in humans. So this is just a first indication of how one could use these tools for studying comparisons between species. So the last chapter I want to talk about is something that just appeared as a preprint on the bioRxiv in the last month and a half, something like that. So this is really hot off the presses. It's the first time I talk about it in public. So we'll see how it goes. But it's really to kind of come back to why these simplices are important, what they're telling us, and what they're telling us about brain function. So trying to make a connection between the complexity of a network as expressed in terms of the simplices, its reliability in always, producing the same output in response to a given input, or a not too different output compared to input, and its efficiency. Because there's this tension when you're thinking about a network of neurons. Your brain has to be fed. It takes a lot of energy to make your brain work. And if you have so you want to be efficient. At the same time, being efficient can be sort of contradictory with being reliable. So that's what we're going to explore now. So what we're going to do is look at 4 different biological neural networks. So there's one that's long been understood, which is the network of C. elegans, the flatworm. So it's very small. It's a very simple animal, has a very simple life, doesn't do much besides moving back and forth and eating, and that's about it. And so there's a full reconstruction of its entire brain of 324 neurons and roughly 2,000 connections. So this is something that is very well understood, and we can apply exactly the same tools I've already talked about. We can do it as well for Drosophila larvae. So this is much more recent, that we have the entire connectome for a Drosophila larva, a fruit fly larva, with roughly 2,700 neurons, and now roughly 64,000 connections. So this is a lot bigger, but still, it's smaller than what we had for the blue brain. And then there's microns. This is also very recent. So in this case, we have a little fewer than 10,000 neurons with roughly 500,000 connections. And we're looking at an electron microscopy reconstruction of now it's mouse visual cortex. So now we're into mammals. This is a lot more complicated. And this is not a reconstruction done by computer. This is really a piece of brain tissue. And then the Blue Brain Project take part of the somatosensory cortex model that I talked about. We have about 27,000 neurons and 6 million connections. So it's roughly the size of the microcircuit we were talking about before. So we can look at all 4 of these networks and do the same kind of analyses. And we're going to compare them to various control models, including the Ardèche-Régnier, so the random graph, where we can maintain the overall density of connections. We can make a directed configuration model where we maintain sort of the number of the in and out degrees. We can look at something that's distance dependent, where the density of the connection depends on the distance between the neurons. And so we have some sort of formula like this with coefficients that we obtain by doing some coefficient fitting with the graphs that we have. We can say, okay, we preserve the underlying undirected graph, then choose the directions on the edges randomly, and then add in the reciprocal connections. And the last control model is to say we maintain everything about the directed graph except where there are reciprocal edges, and just put the reciprocal edges, the bigons, in randomly. And so these are the different control models that were taken. And then how are we going to compare the networks? One way we do it is by saying, well, what's going on with these reciprocal connections? So in the Blue Brain model, for example, of the roughly 8 million edges, there were about 90,000 that were actually reciprocal. So you have A to B and B to A. And so I want to see what is the density of such edges. And what we'll say is, we're going to say that the simplicial weight of an edge, this is like going back to what I was saying about the meaning of simplices, it's the maximal dimension of a simplex containing the edge. And then what we can do is look at the subgraph of the graph G with all the vertices, but only those edges whose simplicial weight is at least k. So we'll throw away all the others. And then we'll say that the kth reciprocity of G is the percentage of edges of GK that are reciprocal. And we want to see whether there's something unusual about the distribution of the reciprocal edges in our graphs. We can also try to take a local view of what's going on by looking at different neighborhoods. So if we're given a graph, and we want V to be a vertex of the graph, then the neighborhood of that vertex is the set, it's the subgraph given by the vertex, all the edges that are directly connected to it, then the other vertices at the other end of those edges, and then the edges between those, and that's it. So that's the neighborhood. And what we can say, well, how many vertices are in that neighborhood? It'd be the size. We could say, we can look at the degree distribution of the neighborhood as compared to some control model and measure some distance between these degree distributions to say how far is the degree distribution from being random. We can do the same thing for its simplicial structure by looking at the numbers of simplices, edges, two simplices, and so on for both the neighborhood and some configuration model. And so if we look at these simplest counts, what we see is that here we're doing the comparison for all of the 4 biological networks with the configuration model, Ardèche-Régnier distance, and so on. And we see that, indeed, the simplicial complexity is much higher in the original graph. And so what we can, and indeed, the computed p-values for the significance here, and they are very, very small. So we see that, in that sense, we can definitely say that these are very different from these particular random graph models in any case. And we can also look at what's happening with reciprocity. So that's the second line here. So see how reciprocity changes with dimension. And in the original case, which is the red curve, we see that the reciprocity goes up with dimension, which means that the higher we go in dimension with a simplex, the more likely it is to have reciprocal edges in it. Or reciprocal edges tend to be more highly present in high-dimensional simplices. And this is not the case in any of the random models. So there's something very interesting going on with reciprocity. We can also look on what's going on with complexity in terms of the complexity of, if you look at the neighborhoods of vertices that are part of simplices of various dimensions, you can look at the vertex, and you can look at the degree complexity of its neighborhood, and you take the average of those degree complexities over all that are in that dimension, and just see what happens as dimension increases. And we see that complexity is increasing with dimension as well for every one of the different biological models that we're looking at. And you can say, well, what is the relationship, for example, between this reciprocity and degree complexity? And you see that, there's I think most biologists would consider these are actually pretty amazing correlations from what I've seen, and that there's a very interesting correlation between the degree complexity, so the complexity of the degree distributions of the neighborhoods, compared to the density of the reciprocity. So there's something interesting and significant going on there. And this is true in all the models. And the little green diagrams up there are comparing it with Erdős-Rényi and saying Erdős-Rényi, there's absolutely no, so this really random model, there's no relationship whatsoever between reciprocity and this notion of degree complexity. Now, okay, what we're really interested in is what this has to do with function. So let's talk about the notion of reliability. We'd really like, you want that if you give the same or a similar input to a network, that the output is going to be the same or similar. So this is this kind of reliability. And so the way to think about this is with this, the plot that's up here on the upper left-hand side, where it says repetitions in time. So what you're doing is repeating the same input many times, and then you look at how the neurons react through time. And it's highly reliable if so each one of those little black marks, this is what's called a raster plot. Each one of those little black marks corresponds to when the neuron emits an action potential, a spike. And those are called spike trains. And you're looking for highly correlated spike trains. And that's when you have high reliability. And low reliability is when there's really no similarity between the spike trains. On the other hand, you can think about neighborhoods in the network that are of low complexity, which would be the green case here, or high complexity, so there are a lot of simplices and so on, which would be the purple one here. And what the graph on the plot on the far right-hand side, so we're looking at violin plots, we're plotting the distributions of reliability for, on the left-hand side, in the green, the 100 neurons that have neighborhoods of lowest complexity, and on the purple side, the 100 neighborhoods that have highest complexity. And there's a significant, with a very low p-value, difference between these the distributions of reliability between high and low complexity. So basically, the higher the complexity, the more reliable the network. So what happens when we reduce complexity? So there are various ways we can reduce complexity by removing structure. And so by everything from something that just says, I want to have a uniform probability of connection, which is giving me basically Erdős-Rényi, to something that takes more and more into account the structure. So these are order 1 to order 5 there. And what we see is that if we look at the dotted black line, that would be the original simplex distribution. And then if we reduce complexity to these various degrees, we definitely see the number of simplex counts, that the distribution of simplex counts that changes, but also the reliability that shifts towards very low reliability. So as you reduce the complexity of the network, you're also reducing the reliability. So this is demonstrating that it's actually a causal effect. And then there's a question of how the relationship between the electrical behavior of two neurons is actually correlated. So this goes back to the graph from a while ago. And again, it's showing that as dimension of a simplex increases, the electrical behavior of neurons is more and more correlated. So this is we knew this was the case in the blue brain thing. And so the guess is, again, the different colors indicate where in the simplex you are, at the source of the sink in the middle. And it's just interesting to see that it is also confirmed by actual biological data from an actual mouse. Now, about efficiency. So reliability is important, but as I said, efficiency is important as well, because you don't want to waste energy. We have limited energy resources, and so we'd like to limit our expenses in order to have the best result. So efficiency here is something we're measuring by saying, what is the number of neurons you need in order to transmit a certain stimulus, depending sort of as a function of the size of the neighborhood. So there are ways of calculating this. It has basically to do with its kind of entropy measure. And what turns out to be the case is that the most efficient neurons tend to be in the lower dimensions, as we see here, although there seems to be some sort of increase as we start increasing again. But that basically efficiency is fighting against, in tension with coupling coefficients, reliability, and complexity. So the last of these is really saying the more complex a neighborhood is, the less efficient it's going to be. So we can't, and we already saw that the more complex, the more reliable. So we absolutely see this tension between reliability and efficiency. So, and here is again looking at these 100 most complex and 100 least complex neighborhoods. And if we look at this on the right-hand side here with efficiency, so the green, I remind you, let's have it up there. The green is low complexity, the purple is high complexity, and we definitely see that the distributions are basically completely disjoint between the efficiency of the least complex neighborhoods and the most complex. So it's really illustrating this tension. So let's summarize, because there was an awful lot coming out there. But the messages are important, I think. So for both directed simplices and reciprocal connections, they're both significantly overexpressed in these biological neural networks. And the overexpression increases with dimension. The higher the dimension of the simplex, the more they're overexpressed, and the more reciprocal connections they have compared to what you would expect. And also, distance dependency and having the correct degree distribution gets you part of the way, but it doesn't suffice to give you the full complexity of the network. Finally, that the neighborhoods within the network, so these neighborhoods of the individual vertices, exhibit tremendous diversity in terms of their complexity in there are a number of different ways you can measure you can characterize, quantify the complexity of these neighborhoods, and there's always a very broad distribution and interesting distribution there. So what does this have to say about function? Well, we observe that lower complexity leads to higher efficiency, and that higher complexity leads to greater reliability, which is good, but lower efficiency. So, and another thing that I didn't really have time to talk about in any detail is that the computations that were done, the simulations that are done, show that efficiency is really something that's happening very locally, whereas reliability is sort of built globally out of these local bits. And so it seems to be important that when you're going to have a biological neural network that's going to function in a good sort of balanced way for an animal, you need this sort of diversity of local structures, the diversity of the different neighborhoods, in order to have a correct structural foundation to enable the functional diversity that we need. And with that, I thank you very much for your attention, and we'll be happy to answer any questions.